Thank you for coming. We are going to talk about adaptive immune system. And probably I don't need to give you a very deep introduction in immune receptors. And T cells and B cells, you know, it's integral part of adaptive system, the most important components. And specifically, we are going to talk about receptors for these T and B cells. And here shown the structure on the left side, you can see in the circles, structure of T cell receptor, B cell receptor, and antibodies, which is soluble form of B cell receptor. They organize very similar way, having light chain, and the sequences which show in the circle, CDR3, recognize foreign antigen or peptide in the context of full-length protein, or as a peptide in the context of the HLA complex. This system is really amazing genetic system created by nature. There are complexity of the system, it's enormous. So there are several hundred million of T cells and B cells with different receptors present in our body. And if you think about it, what does it mean, several hundred million? At least 10,000-fold more than the number of the human genes. So this huge genetic system requires somehow to study. So the first question is very practical. Can you sequence all these hundreds of the millions of receptors and try to do quantitative analysis to identify something which is upregulated or downregulated? And this one, technologically, it's interesting. And our presentation will be some sort of the training or introduction and technologies, how to do it. Second, from biology point of view, adaptive immune system, it's universal diagnostic system created by nature, which allow you to practically identify any pathological condition or any foreign antigen which present in our body. And practical question, why we are not using this universal diagnostic system, which is probably many orders of magnitude more powerful than anything what we developed right now for diagnostic. The answer is very simple. We don't know how T cell receptor or B cell receptor will interact with the antigen. So if you rather easy can sequence immune receptors, link between receptor and peptide, for example, T cell receptor and peptide, which is recognized, is still completely mystery. And it's really a lot of efforts currently done by many research groups in order to decipher this type of interaction. But right now we're at the stage where we don't know really very clear connection between sequence of immune receptor and antigen what they recognize. In order to facilitate these studies, we developed three technologies what we are going to discuss today. And first, it's bulk profiling. So it means if you have RNA or DNA, you can really do immune repertoire profiling for T cell receptors and B cell receptors. And it's rather useful in order to do screening of activated immune receptors by peptides or vaccine, for example. We also developed very similar technology in the middle, shown in the middle, which is single cell analysis. It's some sort of the user-friendly adaptation of the 10x genomics, but instead of encapsulation of the cells in the droplets, you just deposit the cells in the 96 well plate using FACS. So it's very simple. You sort the cells in 96 well plate, and each well you have primers which allow you to amplify immune receptors and markers for the T cells or B cells. And last technology what we are going to discuss is artificial Jurkat K562 reporter cell lines, which is a rather traditional way in immunology to study peptide TCR interactions. And we try to see how these technologies can work in order to identify functional interaction between T cell receptors and peptide. So first, it's bulk profiling. So this one is probably the most comprehensive assay available right now on the market. And you can start from the DNA and RNA, or even you can start with the fusion constructs between human and mouse. Like in the case of when you study, for example, do profiling in mouse cells. And you can amplify CDR3 portion by using the set of the primers designed for C region and FR3, or you can amplify full-length region if you design primers for FR1 region. And what good in this technology, you can amplify all 7 chains if you like to do comprehensive adaptive immune repertoire profiling, or you can amplify only T cell receptor or B cell receptors. Technology work for a wide range of samples. It can be normal RNA or DNA purified from bulk tissue or blood samples, or it can be exosomal RNA or cell-free DNA, or it can be FFP or DNA and RNA samples. Quantitative analysis facilitated using validator barcode. It's concept similar to UMI, which allow you to do error corrections and normalization of all read number to the number of the molecules which used in the reaction. And data analysis done MIXR and or platform, a new version of this software, which is the most popular platform available right now for clonotype analysis identification. And this one is really free for scientific community. I encourage you really to use this platform. It's really cool. It's worked rather well. It's not technology what we developed. It's not multiplex PCR. If you just, we are talking about technology, we use definitely mix of gene-specific primers, but we are using them only in two rounds of extension, like shown here on the top. First, you extended reverse primers, after reverse mixture of forward primers, but amplification done using universal anchor primers. It's guarantee uniform amplification of different fragments for different isoforms of immune receptors and make this assay more easy to quantitate. And after that, it's great. What's good in this technology, it's work not only in purified RNA and DNA. You can even use cell extract, because first step, it's hybridization between primers and nucleic acid. And this one, it doesn't require pure RNA, for example. You can use cell extract, for example, mixture of the cells. Single cell analysis, as I mentioned, is similar platform. In this case, what you're doing, logic here, when you just do, for example, activation of the T cells, usually, or study interesting population of the cells, usually this population of the cells extremely small. It can be maybe a couple hundred cells. In this case, you don't need to run 10x genomics. In any case, you're analyzing these cells by fax. During analysis, you decide, okay, interesting to look more deeply, what are these cells? You sort the cells in 96-well plate. Each well has primers designed for the immune receptor, specific immune receptor, T cell or B cell, and 30 markers, which are top markers for immunophenotyping of the T or B cells. You take the plate, keep it at -80. Refrigerator, when you are ready, you just incubate this plate for 1 hour at 60 degrees, and after that you can mix content of all wells together and run as one sample and to get all information, full-length chain pairing information and expression of 30 markers. Let's look at the data, because it's not about technology. I can talk about technology a lot, but let's look at the data, because it's give you better flavor of what you can do and what you cannot do. And first question is, can we do quantitative analysis considering this enormous complexity of immune receptors? And here's a typical example. For example, you take PBMC or whole blood sample, 50 nanogram RNA, run this assay, and you get approximately 100,000 clonotypes for the T cell receptors and for the B cell receptors, all 7 chains. The question is, you repeat this experiment 3 times. Are the data going to be reproducible, clonotypes what you identify in 3 experiments? And simple answer, it's no. They are not reproducible at all. And this one slide illustrates why they are not reproducible. If you see on the y-axis, it's starting number of the molecule templates for the each clonotype what you identified. And we identify here 100,000 clonotypes. On the x-axis, you see it's number of the clonotypes. If you see majority of the clonotypes, at least 99% of them, amplified only from one molecule. Definitely, if you amplify from one molecule, you cannot really get any reproducible results. It's missing or present. It's some sort of them statistically impossible. But if your starting population is 5, 10 mRNA molecules, in this case, data start to be reproducible, as you can see, in triplicates. And number of these clonotypes which are really reproducible, it's approximately 1,000, just briefly. It's just plus minus, yes? So for definitely this 1,000 clonotypes, where you have number of template molecules 5, 10, you can get reproducible data. And this slide is illustrated. You see here's different amount of starting RNA, even dried blood samples, 30 microliters of blood. You can see it's really data reproducible, and reproducible for both RNA and DNA, like shown here on the bottom. DNA definitely require more number of them. It's micrograms of DNA in order to get reproducible result. Other case, copy number is not enough in order to get any reproducibility. RNA, 50 nanogram, PBMC, it's okay. For tissue samples, definitely you need to use more because content of the lymphocyte may be very low in the tissue samples. The question is, okay, you identify thousands of interesting clonotypes. What you are going to do with these thousands of clonotypes? If you have laboratory with 100 postdocs, probably you can study all of them. But if you don't have these resources, you need to concentrate on something which is important. And definitely the most important, clonotypes which are associated with any disease state or activated by, for example, in any biological process or in your experiment. And one of them, I just concentrate on something what is interesting. And here I just look at the ACR, it looks like, I don't know, 10% of the people definitely doing cell-free DNA analysis, mainly with the focus for mutation analysis, alternative splicing, or just determine the size or methylation pattern of this cell-free DNA. You can do very similar assay if you work with the cell-free DNA and RNA. In this case, you do immune repertoire profiling. And why it's interesting? Because many publications right now, it's some sort of a new frontier in research, and many researchers believe that this fraction of cell-free DNA reflect composition of tumor-infiltrating lymphocytes in the tumor sample, yeah? And genome cell-free DNA reflect apoptosis of T and B cells. So it means that T cells, you can identify T cells and B cells which are dying by cancer, which down-regulate all these cells and try to kill them. At the same time, cell-free RNA fraction, which mainly derived from exosomes, reflect the population of T cells and B cells which are activated and probably fight against cancer. Combination of these two profiling allow you to identify small set of clonotypes, as shown here on the right. It's maybe 20, 50, or 200, or maybe 1,000 clonotypes which already can be good candidates, just to see action hero which are really fight against cancer or dying by the cancer in the course of. And definitely it's important really apply these studies for longitudinal studies. In this case, you can really to see progression of how different T cells and B cells proliferating or dying in the course of, for example, surgery or immunotherapeutic treatment. So here's another example. Here's the vaccination studies. And what we've done here, we just try to see, can we quantitate expression of different receptors after vaccine? And I was actually guinea pig in this experiment. So we collected a lot of time points as shown here for the two vaccines. First of them is triple vaccine, tetanus, diphtheria, and pneumonia. And another one is Shingrix vaccine, second one. So we did expression profiling and adaptive immune and error assay for these samples. We just try to do expression profiling, and this one assay was available from our company. It's profiling of 19,000 protein coding genes using similar concept, using a set of the primers designed for 19,000 protein coding genes, and you can get expression profile. You see, after 2 days after vaccination, you can see really activation of innate immunity pathways on the top, and approximately 1 week you get activation of adaptive immunity pathways. If you try to do RSA for the same samples, you can see here in the center you can see B cell responses, and it's IgH, it's heavy chain of immunoglobulins, and you can see here it's at the same time, approximately 1 week after vaccination, you can see strong activation, approximately 100 of different clonotypes for B cell receptor, and smaller number of clonotypes, similar number of clonotypes for Shingrix vaccine. And you can see here it's really very strong effect. It's up to 10,000-fold activation of these some clonotypes. It's combination transcriptional activation and at the same time expansion of the B cells which recognize these foreign antigens of this vaccine. And definitely it's majority of them, it's plasma cells probably with IgM phenotype. So it's interesting to note that T cell receptor is also activated. We don't see significant proliferation of the T cells after vaccination, but we can see transcriptional activation, which indicate that T cells and B cells cooperatively interact to each other in these immune responses. So we try it's interesting. We talked that it's very difficult to predict TCR and peptide interaction. We try to apply this data and try to see. We work with the company ImmuneWatch, and they have very cool machine learning algorithm which allow you to see if any T cell receptor what you identify can find match or the existing database. We did not find any match for the first vaccination with triple vaccine, but for Chenchik vaccine, significant fraction of the T cell receptor sequences shown here are associated with zoster virus glycoprotein E. So this indicates that sometimes this type of the bioinformatics strategies, maybe it's not a bad idea just to test them if you have list of the TCR candidates, just to see them if it's possible to predict what peptide they recognize. So we just discussed that it's really cool, but majority of the people here, for example, they are doing what is the typical TCR antigen interaction experiment. In majority of the studies, you have big list of the peptides which generated, for example, from genome sequencing of tumor samples, and based on using RNA or DNA, with the goal to predict what novel cancer-associated antigens or mutated neoantigens are present in the biological sample. It also can be experimental strategies like shown here, mass spec of identification of peptides which bind to the cancer cells. In the course of all these experiments, you generate a long list of candidate peptides, which can be interesting targets in order to study, to identify matched T cell receptor, and at the same time use them in order to develop novel cancer-specific vaccine or T cell-based therapies. And the question is how to identify matched these peptide candidates and T cell receptor. Classical way to do it, it's really the most simple. It's like published in many articles, and we just go with our research was just reproduce this study and to see how this technology, what I described, can be used in this pipeline. You take PBMC from normal donor or disease donor, stimulate this PBMC with the peptide candidate, what you discover in your studies. And here's the example of MART1, which is one of the cancer-associated peptide. And you can see you grow cells in the presence of some combination of cytokines. And in 2 weeks, you can do analysis. If you compare samples time 0 and 2 weeks, you can see here on the bottom, you can easily identify clonotypes which are expanded, stimulated by peptides and expanded in this pool. After 2 weeks, what you can do re-stimulation of this PBMC again with the peptide and do single cell analysis in order to identify full-length chain pairing information for T cell receptors in order to use them for follow-up validation studies. Here's the result of this experiment. It's heat map. If you look at the from the left, yes. First column, it's original population of PBMC. And second, it's population of the clonotypes after DMSO control treatment. And third column, it's clonotypes which are expanded after peptide stimulation. You can see here it's expansion level of the multiple clonotypes, and here it's approximately 50, several thousand-fold. So it's very clear. See, a lot of these TCR receptors are probably peptide specific and can be used for validation studies. But in order to narrow down this list of candidates, we did staining, as I mentioned, after re-stimulation for CD137 marker, MART1, tetramer, and interferon gamma. You can see next 3 columns. You can see data also rather good overlap between this data and data identified by quantitative air analysis, but it's more sparse. It's just not as reproducible as quantitative profiling data. Last column, yellow column, you see, next one. It's interesting. The same ImmuneWatch algorithm allow you to predict if this T receptor recognize this MART1 peptide or not. And you can see here it's also rather good overlap with the data quantitative analysis using air assay. So we take 12 candidates from these studies and validate them in this system, which is artificial system. And we developed this system. It's rather classical. It's even probably already used more than 20 years ago by Rosenberg from NCI. And this one is based on Jurkat reporter cell lines with the NFAT reporter. And you can clone any T cell receptor in this left Jurkat reporter cell lines. And if T cell receptor activated, it's expressed GFP. reporter. K562 cells is good because it doesn't have any HLA, so you can clone any HLA you want, or peptide in the form of mini gene or fusion between HLA, B22 microglobulin and peptide, or use synthetic peptide just using the K562 with expressed HLA complex. So combination of these cells allow you really to identify if it's functional interaction between peptide and T cell receptor, as shown here on the right of the slide for CMV T cell receptor. So using FACS analysis, where we clone all these candidates, 12 candidates, and use spiked peptide with K562 cells with matched HLA background, you can see from 12, 80% of the clones show very strong GFP activation, which show rather easy passive way to really to do this type of the analysis. The question is, can you use it for a large scale? Probably not, because it will be really a lot of efforts, significant efforts, only 3 weeks. You can scale up maybe up to 10. But if we are talking about hundreds of the candidate peptides or thousands of peptides, it's probably the only option really to do it is just to go to use libraries. And libraries here, you have several options. You can, like shown on the top, you can build synthetic T cell receptor libraries. And right now, with the wake of all this gene synthesis, right now building of T cell receptor libraries, it's not something outstandingly complex. It's rather easy. If you want to build any T cell receptor libraries, we can do it for you. We can build library for 100 or 1000 T cell receptors, which you identify from, for example, from tumor infiltrating lymphocytes. Or you can build peptide libraries. Synthetic libraries can be cloned in Jurkat reporter cell lines. Also, you can work with natural CD8 cells in this type of experiments, purified from healthy donor PBMC samples or from disease samples, patient samples. Peptide libraries, synthetic peptides libraries, even more simple to build, and you can make them in definitely K562 as the antigen-presenting cells. You can co-culture these cells in this platform shown here. This one is the most simple way what we find how to study this type of the interactions. And you see core of this technology, it's 96 well plate. In each well, you have 4,000 microvels. Size of these microvels slightly bigger than cell. So what you can do, you can deposit antigen-presenting cells, one T cell, and each well, microvell covered by antibodies. Antibodies recognize, for example, interferon gamma, depending on the USA, but this one is the most popular. It's similar to what people use in ELISpot assay. If these cells are interact to each other, they release interferon gamma. Interferon gamma can be detected using detection antibodies, which constantly present in the media above these microvels. In order to do this analysis more efficient, here's the instrument what our collaborator company CellDome is using. This one's Clone Explorer, and this machine what is doing, it's doing quantitative online analysis. You can incubate plates in this instrument up to 2 weeks, because it's also at the same time CO2 incubator. And it's see every cell what you deposited, antigen presenting cell and T cell which you deposited in microvell. It's monitor how they proliferate in time. Also at the same time it's measure fluorescent signal generated by detection antibodies, or if you use Jurkat reporter cell lines, it's measure GFP expression level. And what good in this machine, after identification of these positive cells, it has a micro syringe which allows it to collect all cells from each positive cells and transfer to 96 well plate in order to analyze composition of T cell receptor what present in this positive well and peptide which is activate T cell receptor. And we done this experiment using this technology. We done the experiment by building library of 120 peptides, clone them in HLA-02, HLA-0201 background as the trimeric complex, and screen them using CMV construct cloned in Jurkat reporter cell lines. You can see here, here's the original population of the peptides in the library, and here what's happened when we select GFP positive clones. Majority of clones what we selected has CMV peptides, which indicate how this technology, demonstrate how this technology can be used for this type of the screens. So we discuss really combination of several technologies in order to identify T-cell antigen interactions. And here's the summary of what we discussed today. We discussed development of quantitative air profiling assay. We discussed a strategy how to do single cell analysis for small number of the interesting cells using sorting of the cells in single cell air assay. We discussed strategy for screening of TCR antigen interactions using synthetic peptides and ELISpot or fluorospot microwell technology. And if you're interested to use this technology, all of them are available as the products or custom service. And if you're interested, contact us. It will be our pleasure to help you in your research.